Hi everyone. It's 4 o'clock here in the UK. Thank you for joining us for another of the Simcyp scientific webinar series. So today we're honoured to have Professor David Taft. And David, if you could just put on the next slide, please. Thanks. So David received his bachelor's in pharmacy from University of Rhode Island, and he then completed a Ph.D. in University of Connecticut. Since then, he completed a fellowship in pharmacokinetics, University of North Carolina School of Pharmacy, and he joined Long Island University in 1994, serving as Dean of the College of Pharmacy from 2008 to 2013. So those of you who know David, he's a big advocate of teaching, and he teaches a lot of courses, particularly in pharmacokinetics, in both I believe there's a number of programs at Long Island University that he teaches on. And in terms of his primary research area, it is pharmacokinetics. And so he's been working on projects ranging from characterizing renal clearance or renal excretion in rat kidney models, looking at drug formulations in preclinical and clinical studies. And he's won a number of awards, including a ACP New Investigators Award. And again, you can see an award for teaching. I'm really glad that he agreed to talk to us today. He has been a winner on the grant partnership scheme at Simcyp, which he's presented in another webinar. But this presentation relates to another piece of work, which is a recent publication, and it's PBPK modeling of altered Tizanidine exposure by CYP1A2 modulation. So this should be a really interesting case study. It's got really complex mechanisms in terms of different drug-drug interactions, and also considering the effect on cigarette consumption. So I'm really looking forward to David's talk. So I'll hand over to you now. Thank you, David.
Well, thank you for the introduction, Ali, and hello, everybody. Yeah, my career has been primarily, I've been in pharmacokinetics, I would think the last 5 years or so since I became involved with Simcyp. A lot of the research that we're doing is really based on PBPK modeling. Students really enjoy that, and they've really gotten a lot out of it. And so today's talk, I'm giving credit to the first person here on the screen. This is Vamshi Krishna Jogaraju. This was Vamshi's part of his PhD project. Vamshi now, he graduated about 1.5 years ago. He now works for Gilead Sciences, but he designed the project, he executed it, and so even though I'm presenting this here today, this is really Vamshi's, at least a piece of his thesis work. And so the idea with this was finding a case study example compound or medication, which is Tizanidine, and really going through and relying or using PBPK modeling with Simcyp to really look at how we can develop a model to probe altered modulation of CYP1A2 and how that affects Tizanidine exposure. And so I realized that most of you in the audience here, are very familiar with PBPK modeling and applications of Simcyp, particularly on the industry side. So I'm not really going to spend a lot of time with some of these introductory slides, but obviously the goal in drug development is to have safe and effective drug therapy. And we're aware of patient factors, whether they're intrinsic factors or extrinsic factors, that can alter the pharmacokinetics of a drug. And as a result, that will alter the systemic exposure and could result in changes in safety and or efficacy. So the real goal today is to focus more on the intrinsic factors, and specifically with CYP1A2, certainly there are perpetrators in terms of drug-drug interaction perpetrators, but as you know, and as somebody who's a pharmacist, when I was in pharmacy school back in the late 1980s, a common asthma medication was theophylline. And it still may be used a little bit today, but back then it was probably the most commonly prescribed, at least oral dosage form for treat asthma. And it was well known that theophylline pharmacokinetics or clearance was dramatically different in smokers. And so in this particular case study, we also wanted to probe some environmental factors, particularly smoking. So here is the general setup. And again, people do know this. So I apologize if this is, something that you're well aware of, but the typical language, we refer to victims and perpetrators for metabolic drug-drug interactions. So obviously transporter-mediated interactions are also very important, but particularly for this case study, focusing more on the drug metabolism side, that we have our victim drug, and if you have potential co-administration of medications that are perpetrators, they can either induce that enzyme, and resulting in increased transcription, higher enzyme abundance, and increasing the intrinsic clearance of the drug. That could potentially, without dosing adjustments, result in reduced systemic exposure, and then you wind up with potential loss of efficacy. On the other side, if your perpetrator is an inhibitor, it works the other way, that you have reductions in clearance, increased systemic exposure, and run the risk of potential toxicity. What you'll see today with Tizanidine is that is a real threat, because the sensitivity of Tizanidine to this particular pathway is really important. So just to review the general types of interactions that we have, the most common ones are certainly inhibition and induction. Again, I'm not going to spend a lot of time on these, and you'll see these in the case study. But with competitive inhibition, the inhibitor is competing for the enzyme at the same time as the victim. And the two important characteristics are relative concentrations of the perpetrator and the victim, but also the potency, the inhibitory potency of the perpetrator for the enzyme. And so that's what's shown here on the left-hand side of the screen. And what we use, when you look at this general scheme, this is where the Michaelis-Menten equation comes from, and our understanding how intrinsic clearance relates to Vmax and Km. When you look at the bottom here, the other type of important inhibition is the time-dependent or mechanism-based inhibition. And that's really modeled based upon natural enzyme turnover in the body, is based on a zero-order production, which is the synthetic K-syn, and a first-order degradation process related to the enzyme level itself. In the presence of a mechanism-based or inhibitor, what happens is that inhibitor binding to the enzyme actually increases the degradation of the enzyme. And as a result, over time, enzyme levels fall. And so when you look at the important parameters that you have to model with mechanism-based inhibition, it's really this K inactivation and also Ki. And then on the other side is when you actually increase enzyme expression. This is where you have the induction effect. And what you see here is it's really based on an Emax model, where you're really looking here at what's the maximum inductive effect, as well as the induction C50, which is similar to an EC50. Now, in drug development, we have these static predictions, which are commonly used, that can be used to give some indication of the severity of a drug-drug interaction. And so what's being shown here in these boxes are, for each type of potential interaction, these are the AUC ratios. I will point out here that there's a more general form of the equation that takes into account the fraction by which the specific pathway being inhibited or induced contributes to the overall metabolism of a drug. What's being shown here are these predictions when you have more of a single pathway. And that's really the example with Tizanidine. I would point out that static predictions sometimes aren't very reliable. And the reason for that is that when you look at the concentrations of the inhibitor here, they're held as a constant. And they're typically related to, for example, the expected Cmax, plasma Cmax of the drug. And as a result, they can sometimes overpredict the magnitude of the interaction, because ideally you'd like to have more of a mechanistic or dynamic way to probe that drug-drug interaction. And that's where physiologic-based pharmacokinetic modeling comes in, that you're able to rely on probing or evaluating changes in the concentration of your victim substrate and your perpetrator over time, not only in the plasma, but at different organs and tissues in the body, including the liver, where drug metabolism takes place. And mechanistically, you can probe perhaps more real expectations of what the magnitude of the drug-drug interaction is. So again, I'm not going to spend a lot of time here because I think everybody here is familiar with how PBPK modeling is done, but obviously we have a strong relationship with Certara and Simcyp, and we're very fortunate through the generosity of Certara that we're able to use Simcyp in our academic pursuits. And certainly this is a model that has been proved, or a platform that's been proven to be very important in drug development and helping to accelerate the drug development process. So just to give an idea, this is an article that came out, I think, in 2019, but it's based on data collected over a 10-year period by the FDA. And what it shows, it demonstrates the importance of how PBPK modeling is being used in drug development and in regulatory submissions. And so it's pretty striking that you have over 200 submissions, whether they're INDs or NDAs, where PBPK modeling was included in those submissions. And at the time, anyway, based on that evaluation, DDIs, drug-drug interactions, were the predominant role of PBPK modeling. I think if you look at the literature since 2018 or more recently, you see a lot more. There's expanded use of PBPK modeling in special populations, such as pediatrics, looking at virtual bioequivalence, for example, with transdermals. There's a lot of widespread use that I know most people are familiar with that's happening now with PBPK modeling, but it still is very important for DDI probing. So the example today, what we're talking about is a molecule where obviously a lot of the emphasis, at least in terms of cytochrome P450, has been perhaps more on 3A4, perhaps 2D6, 2C9, 2C19, and for obvious reasons, given the importance of those pathways for medications. But the CYP1A2 pathway is interesting as well, and it has some interesting substrates, many central nervous system acting medications, antidepressants, antipsychotics. You see here theophylline and caffeine. Caffeine is generally used as a probe for CYP1A2. I mentioned theophylline before as a substrate that I'm familiar with. CYP1A2 is highly variable, and that's due in part to modulation, drugs and chemicals that induce or inhibit activity. It has some interesting things like cruciferous vegetables, like Brussels sprouts and broccoli can modulate CYP1A2, but perhaps the most, one of the most important environmental concerns or factors is certainly smoking. Smokers generally have increased CYP1A2 expression. And the molecule that we chose, or the drug that we chose in this case study, is an interesting one as well. This is Tizanidine, which has a couple of brand names, but in the US goes by the brand name Zanaflex. And this drug works, is used to help patients with either multiple sclerosis or spinal injury when they deal with spasticity. So it's a centrally acting alpha-2 agonist. As far as its pharmacokinetic properties, what makes it a little bit interesting, particularly, is when you look at the two factors in this bullet point is that it's primarily cleared through hepatic metabolism. There is a small contribution of renal excretion to Tizanidine, but it's an example of a molecule where it really has one way out. There's one way that it gets cleared from the body, and that's really CYP1A2 mediated metabolism. The estimated fraction of CYP1A2 to the overall metabolism of Tizanidine is close to 100%. And so, as you could probably expect, is that when in the presence of a potential perpetrator for 1A2, you would expect to see tremendous differences in systemic exposure with Tizanidine. And that's what makes it an interesting molecule. It also has relatively low bioavailability. I don't think 40% is necessarily low, although it is variable. And the primary reason for that, it has good absorption, it has good stability in the intestines, but the main reason for its bioavailability of 40% is due to pre-systemic hepatic metabolism. So it's a relatively high clearance drug, and that clearance is primarily mediated by 1A2. And it is a sensitive index substrate. The general guideline is if a substrate shows a 5-fold or greater increase in AUC ratio in the presence of a strong inhibitor, that would be a sensitive substrate. And you're going to see tizanidine is definitely a sensitive index substrate for 1A2. So as I mentioned, this was a part of Vamshi's Ph.D. Work. So we were concerned or interested in model-informed drug development. And so this research also probed into examples of using PBPK pharmacodynamic modeling to look at how elevated exposure may affect safety. There was work done on special populations like pediatrics and patients with renal or hepatic insufficiency. But to speak today is really introducing the model that Vamshi created, and then show how we used drug-drug interaction or CYP1A2 modulation studies to demonstrate application of the model. So specific aim 1 is really just to develop that model and to verify that based on available clinical data. And then specific aim 2 was looking at these intrinsic factors, co-administration with other medications that may modulate 1A2, and also including the impact of smoking. What made this case study available to us is the availability of published data on tizanidine. There's a number of clinical studies that were published by back in the about a decade and a half ago, looking at tizanidine drug-drug interaction studies in humans. And so we took advantage of that available data to help inform the development and the application of the model. So in terms of methodology, I just took this from the guidance that came out about a year and a half ago on the in vitro interaction studies. I'm sure you're familiar with that guidance, this table is shown there. And this is generally the approach that we used, more of a middle-out approach in developing the tizanidine model. We also developed a model for one of our perpetrators, and you'll see it as Rofecoxib, but we based it on available information from human clinical studies in addition to available physicochemical data and other information that we had. We developed that model. We refined it as needed using parameter estimation and other tools to wind up with a final PBPK model. And then we probed different scenarios with different types of impact of CYP1A2 modulation, whether it was through competitive inhibition, mechanism-based inhibition, or enzyme induction. So we simulated the interactions, we evaluated the exposure differences, and we're able to compare those against published data. And hopefully what you'll see is that our model was fairly well predictive of the impact of CYP1A2 modulation on tizanidine. And we also did do, and I'll mention this as we go along, some sensitivity analysis to try to understand if there was some model uncertainty and try to find a reason for that. So here is our flow chart, and this is how I'm going to walk you through this, that we're going to start with showing you the model and then go through the examples of how we probed all of these different pathways or potential interactions. And these are the perpetrators that we used in addition to looking at smoking. So specific aim number 1, and this is really the smaller aim because we're just introducing the model itself, is the tizanidine PBPK model. So what's shown here, just as a starting point, and I guess in terms of model development, sometimes having a lot of information can pose a potential problem, but this is data for a 4 milligram dose of tizanidine. I think there's 5 different publications here. These were the drug-drug interaction studies that I mentioned before. And what you see is that there is some variability in the mean plasma concentration time profile for tizanidine across those clinical studies. That's not unexpected. These were relatively small number of subjects in each study, but in terms of model development, obviously we can't find one model that will fit all of these data to the same extent. And so our approach was to rely on one of these studies, particularly that looked at the interaction between tizanidine and fluvoxamine, and then applied that model looking at the other drug-drug interaction studies. So here are the properties, or the how we came up with the model. And again, this information, I'll show you the reference of the publication at the end of the talk, but you can find that information in the publication. We did use a full PBPK model with the atom option and absorption. The one thing to point out is that we use the retrograde calculator to estimate intrinsic clearance from the reported oral clearance of tizanidine in humans. And that's how we came up with this intrinsic clearance value. We assigned all of the enzyme kinetics, all of the clearance to CYP1A2. So it was about, I think in the research, about 98%, fraction metabolized is 0.98. We attributed all of the metabolism to tizanidine. So here is the model verification. This was based on this particular reference, this Grandfor's reference. The study involved 10 healthy volunteers. So we ran our simulations. We did replicates, a 10 by 10 in this particular case, study design. And so if you look at the figure here, I think you'll see that we have fairly good agreement between the model simulated solid red line profile for tizanidine, and then the blue circles are the observed data from that Grandfor study. Also, you'll see down here the observed and predicted Cmax and AUC, and again, based on the fold errors, are fairly in line with the observed data. So again, this is the model, the verification of the model for tizanidine. So moving on now, and we have all of these other clinical data available to us, that we wanted to start to probe now different ways that CYP1A2 could be modulated and how that would affect tizanidine exposure. So starting first with competitive inhibition, we did rely on Fluvoxamine and Ciprofloxacin. They are both available in the Simcyp compound library. And so I'm going to start there and show you what our results were with competitive inhibition of tizanidine. So the first perpetrator that we looked at was fluvoxamine. And so as we went through the modeling process, and it really came down to when we submitted the work for publication, the reviewers requested that even though we were relying on compound, the library, Simcyp library compounds for perpetrators that were verified before being put into that library, we were asked to demonstrate verification of the model, even though it was already within the Simcyp library. And so what I'm showing here is this is the compound file. So you can find this in the Simcyp library. The one thing to point out here, when you look at fluvoxamine, it's a very potent CYP1A2 inhibitor. It's got a Ki inhibition constant of around 2 nanomolar. So it's really a strong inhibitor. But anyway, we did verify the fluvoxamine model by looking at comparing it to published data, and we think we're able to capture the systemic profile for the perpetrator. And then what we also looked at are published studies with theophylline, which is a CYP1A2 substrate, and we evaluated the ability with fluvoxamine as the inhibitor against theophylline. And what you see here again, fairly good agreement between the observed and clinical data observed AUC ratio compared to the model predicted values, and the same thing for Cmax. So here is moving on now to particularly looking at tizanidine, and this is a few things to say here about this particular slide. First of all, here's your study design. So this was that Grandfor study where fluvoxamine was given as 100 milligrams per day for 4 days. And then on day 4, tizanidine was dosed as a single dose, 4 milligrams. And so what you're looking at here is time beginning on day 4. And so a couple of things to point out. that the red circles represent the control data. This is actually the same data we used to develop the model. And then the green triangles here represent the interaction, tizanidine in the presence of tizanidine. Down here in this box, this is specific. This is showing only the values for the green, the interaction phase. And what you see is that we were able to capture that fairly well. The big thing to point out here, and surprisingly, I haven't seen an interaction this dramatic, but the AUC ratio is over 30-fold. So you're looking at a situation here where if you administered to a patient tizanidine with fluvoxamine, according to this particular scheme, you're looking at a 30-fold or more increase in AUC, which is why it's not surprising if you look at the tizanidine label that tizanidine is contraindicated with strong CYP1A2 inhibitors. So we have a very sensitive substrate in tizanidine for the CYP1A2 pathway. Now, one other thing to look at here, and again, I apologize if those of you that are active users of Simcyp are very familiar with this, but the Simcyp output allows you to probe, mechanistically look at the interaction. And so on the left-hand side, top left, this is the simulated systemic profile for fluvoxamine. So as a once-a-day dose, what you find is that there is some accumulation up to day 4, but the key point is that since the magnitude of a competitive interaction does depend upon the relative concentration of the perpetrator at the site of the interaction in the liver, that you do have maintained exposure over the course of therapy. But as a result, what you see is a dramatic reduction in intrinsic clearance of tizanidine, almost goes to 0 for that pathway. The reason for that is you do have exposure of tizanidine, of fluvoxamine throughout the dosing interval, but also that inhibitory constant, that Ki value, it's such a potent inhibitor, it essentially wipes out the pathway. And again, as I testified or mentioned previously, that when you have a medication where it's basically a single pathway through which it gets predominantly cleared, and you inhibit that pathway to a strong degree, the end result is you're going to have dramatic increases in exposure. I didn't mention this when I showed you the profile that was set up, but the renal clearance of tizanidine is around 7 liters per hour. So even though the kidney plays a relatively small role in the overall excretion of the drug, it is able to, in the overall clearance of the drug, there is some contribution of the kidney. But as you can see here, you essentially are knocking down almost the entire intrinsic clearance. And notice here as well, the in vivo intrinsic clearance value is very high. And that correlates with a relatively large clearance of tizanidine. And as you would expect, the interaction is a competitive one, so there's no impact on the CYP1A2 expression in the liver, which is what you'd expect with competitive inhibition. Since we also had available in the library to us ciprofloxacin, we decided to probe ciprofloxacin as well. Ciprofloxacin is also contraindicated with tizanidine. It is considered a strong CYP1A2 inhibitor, but I think the way we describe it is moderately strong because it's not as strong as fluvoxamine. And you can see that when you look here at the substrate profile for ciprofloxacin, its Ki value still relatively potent, but it's 1,000 times less potent than fluvoxamine. And so this was our second competitive inhibitor that we decided to probe. Here are the details on the compound file that you can find within the Simcyp library. Again, we did do some verification that we simulated the PBPK model to look at systemic exposure of ciprofloxacin. And here, you see the values. And likewise, we also wanted to verify CYP1A2 modulation with theophylline because there's clinically published data available allowing us to do so. So again, now we've done that step of verifying the internal Simcyp model, and now looking at our drug-drug interaction. This is another study by Granfors, which is a separate study than the one that was used to probe fluvoxamine. And so what you see here is, again, the simulated and the observed data. And you may be saying or looking at this and saying, well, it doesn't appear that our model is completely capturing the observed data. And if you look back to the slide I showed at the beginning, that's one of the issues that we had in developing the model, is that we had data from 5 different studies where there were differences in exposure. And so we used the same model for all of these simulations. And so we do recognize that. But what you see here, we still were able to capture the interaction data fairly well. I would say that, and again, before I go on, the AUC ratio, the observed value was around 9 or 10, slightly less for our predicted value. It's still showing what you see is a significant interaction with Tizanidine and Ciprofloxacin. It is a contraindication. And when we looked at our model, what we wanted to understand is why is our model slightly underpredicting the drug-drug interaction? So we did do a sensitivity analysis. And again, if this was the only interaction that we were working off of, in other words, if we were just looking at this one study, we obviously would have improved the model for Tizanidine to better capture these green circles. And the way to do that would be to improve or optimize our intrinsic clearance value. When we actually looked at, because obviously this would be consistent with maybe in this group, a faster, more rapid intrinsic clearance. When we ran a sensitivity analysis to look at, for example, how AUC ratio changes with intrinsic clearance, what we found is that as intrinsic clearance increases in the sensitivity analysis, the magnitude of the AUC ratio increases as well. And so that at least partially explains why we slightly underpredict the magnitude of the interaction, is because we are slightly over-predicting the Tizanidine levels through the model, because again, our model was verified based on a different Grand Force study. It's still reasonably predictive here, but that would explain, at least in part, the reason for we're a little bit off with the magnitude of the interaction. Moving on to the other type of potential interaction or inhibition is mechanism-based inactivation. And so what we relied on here was published data with Rofecoxib. Vioxx was the brand name. It was removed from the market about a decade ago, but there is published data about Rofecoxib as being a modulator of CYP1A2, and there was clinical data for Rofecoxib as an inhibitor of not only of 1A2 substrates, including Tizanidine. So that's why we included it in the model. And so here is the data that was used to help build the model, because we had to build the model for Rofecoxib. And so again, you're looking over here on the right at what mechanism-based inhibition is. This is the published data demonstrating, for example, Rofecoxib as a time-dependent inhibitor. Pre-incubation makes a stronger inhibition of the phenacetin pathway, which is the index for 1A2. And so from here, we're able to obtain those parameters for the interaction. But we had to first build the model for Rofecoxib. And again, we went through the typical process that people that are building models go through, is look to the literature to get information about the physical chemical properties and other things where gaps existed, to rely on the prediction toolbox as needed. And again, what you see here, we had some in vivo data we could rely on, and then the interaction parameters. So we had to do a couple of steps here. The first step was, can we simulate or capture the observed systemic profile for Rofecoxib? And we did have a study in drug metabolism and disposition with 6 volunteers, where we have the green circles here that show the observed mean data. And as you can see, we fairly well captured that profile, and the data is shown down in the box below. So we feel that at this point, we had a model that was verified. We obviously had to maybe optimize 1 or 2 parameters, but in the process, but that was our verified model. We then moved on to see, we had information of Rofecoxib as a perpetrator for 2 different CYP1A2 substrates. One was theophylline, and the other one was Tizanidine. So this is showing here model simulations looking at Rofecoxib as a mechanism-based inhibitor, the perpetrator, with theophylline as a victim. The study design from the clinical study, the Bachmann study, was 50 milligrams a day of Rofecoxib for 7 days and theophylline administered on day 7. So again, here, this is showing at the beginning of day 7. And so, admittedly here, we were able to capture the control phase fairly well, but the model slightly overpredicted the magnitude of the interaction for theophylline. And so, again, our fold error is still acceptable, 1.9. But this is at least we're showing a verification here that it is at least, it seems to be capturing mechanism-based inhibition with theophylline as a substrate. When we apply the same model to Tizanidine, these are the results that we get. So here again, just to point out that we do again have slight differences in the prediction of the magnitude of the interaction. That may be due in part again that we're using the model that we developed in the control phase was based on a different clinical study. So our intrinsic clearance value was not optimized specifically for this particular data set. But I think if you look at this, the model seems to capture fairly well the drug-drug interaction. Again, what's shown here in the box is really for the red triangles as far as the observed and the predicted data. But we do think that it does demonstrate adequate description or capturing of mechanism-based time-dependent inhibition of Rofecoxib. So again, here are the AUC ratios, which is, if in this case, Rofecoxib is also, this is a strong interaction with an AUC ratio close to 10. And what's shown here is what I showed before for the competitive interaction is looking at the Rofecoxib systemic profile and looking specifically here now, the key thing that happens with mechanism-based inhibition is that enzyme expression is knocked down. And so that's what you see here, where at day 7, the Rofecoxib enzyme levels have dropped to less than 20%. And as a result, there's a significant drop in intrinsic clearance, and that shows up with an AUC ratio of 10. So again, this is mechanistically how you can use Simcyp to probe or learn or understand why you're seeing that disparity or that increase in AUC ratio. Lastly here, just to walk you through the other aspect of modulation, which instead of knocking down or inhibiting a pathway, is looking at induction. So the last example here is looking at induction through perhaps the most common inducer of transporters and enzymes, which is rifampicin, but also looking at those polycystic aromatic hydrocarbons, the PAHs, in smoking, and how that could also impact enzyme expression. So as you saw before, when we actually submitted this for publication, we were asked to demonstrate that the library file for rifampicin was, we could verify that. I did go back and check this. We actually relied on a published reference to add in the CYP1A2 induction parameters for rifampicin. We used Simcyp version 18. I think today in version 20 or 19 that pathway is accounted for in the compound file, but these parameters came from a published study. But this is the compound file, and we did verify this against theophylline. We had a couple of published studies. And what you see is that, again, the model seemed to capture the AUC ratio fairly well. We did not run a verification of the plasma profile for rifampicin. That was not something we did as part of this work, but that's why I'm not showing that here. But we did run a verification of CYP1A2 modulation using theophylline. And here are the results for Tizanidine. This is a Bachmann publication. You know, it's funny, we have an author, Dr. Bachmann, and then we have Bachmann. They're two different people. So I thought I made a typo in one of my slides, but they are different authors. This is a study in 10 healthy volunteers. Rifampicin was administered 600 milligrams a day for 5 days, and then Tizanidine was introduced on day 6. And so what you see here again, and in this case now, the open circles are the control data, and the green represent the interaction phase. So again, as I've mentioned before, the slight differences between the model predicted and observed data reflect that our model is optimized for a different clinical study for tizanidine. But again, I think when you look at this, you see that, first of all, this is referring to the green circles here, that we're capturing the model fairly well. And we actually look at the AUC ratios, they're comparable, that the Simcyp predictions capture what looks like a reduction of AUC of roughly, an AUC ratio drops from 1 down to 0.5, which is reduced exposure in the presence of an inducer. And then lastly, well, one more point here, to be consistent with the other slides that I've shown, looking mechanistically at how enzyme expression changes. In the case here now, we're looking at enzyme induction. So it's increasing from baseline. The active enzyme level essentially doubles. It goes up to about 200%. And as a result, in the presence of rifampicin at day 7, the intrinsic clearance essentially doubles for tizanidine. And as a result, what you see is an increase in systemic clearance, but also an increase in pre-systemic metabolism, and that results in that reduced AUC ratio. So lastly, our last modulator is not a drug per se, it's an environmental factor, and it's cigarette smoking. And so in building this model, it really relied on, and I think most people know Karen Rowland-Yale and Dave Plowchuk from Pfizer, that they had a publication back, I don't know the exact year it was published, but they were able to provide this table of CYP1A2 abundance as a function of how many cigarettes a person smokes a day. So the more smoking, the higher abundance. It was really based on this reference here. That was a study that actually looked at caffeine clearance in smokers and non-smokers, that the authors relied on this study to generate that table. So now we're able to go in and modify the population for different levels of smokers and see how that affects tizanidine. And so we actually probed all of those different levels of smoking, and I didn't provide the slide, but you could see that with people that have a greater frequency of smoking, that tizanidine levels actually decline if you do a comparison of the graph. But what we're showing here is that looking at, in this particular study, again, another Backman study, the tizanidine in the presence of smokers that were in that 10 to 20 cigarettes a day versus non-smokers. So again, here, the open circles represent the non-smokers. The green triangles are the smokers themselves. And what you see, though, is that the model does capture, an impact of smoking on exposure. So again, the data shown here are the observed and predicted values for the actual smokers here. So the key point is that another advantage or application you can have with a drug that's a substrate for CYP1A2 is you can actually probe smoking. You can go into the population page file in your model and alter the CYP1A2 abundance and see how that affects systemic exposure. So just to summarize here, Vamshi created these nice forest plots, which really show the magnitude of the interaction, where obviously if it's a range of 1, that means no interaction at all. To the right, that's increased exposure. To the left is reduced exposure. And so on this left panel, we have Rofecoxib, Ciprofloxacin, Fluvoxamine with Cmax and AUC ratios. And you see how if you look within the observed and predicted, they match up what I think fairly well. And then on the left-hand side, we're looking at our modulation with increased enzyme expression, either through smoking or through rifampicin. And what you see here, again, is the AUC ratios going to the left because we have reduced exposure. So just to summarize, and I think I'm just about right on time, which is surprising for me, but so what are the conclusions here? We're an academic group, so we were interested in a case study exercise from an academic perspective, but we're hopeful that people working in industry may find this model useful. So I think we've demonstrated that we're able to develop a model for tizanidine using the simulator. We have shared the model for both tizanidine and Rofecoxib with the Simcyp team, and I think it is available in the members area if you're interested in it. Hopefully I demonstrated that how we use the model to predict interactions between tizanidine as a victim with different modulators of 1A2, whether they were particular drugs such as Fluvoxamine or Rifampicin, but also looking at cigarette smoking based on those clinical observations. And then we're hopeful. We had a little discussion with Ali, asked me if we've heard back about our manuscript from anybody yet, but we haven't really gotten any feedback on it. But in our view, we believe that this model may have potential application in drug development for investigating drug candidates that may be potential perpetrators of CYP1A2 because it is a sensitive substrate. And so we're hopeful that perhaps there's some utility of this model among the industry. Here is the publication. It is in Drug Metabolism and Pharmacokinetics. And so that's the title page. I could send the reference if you need it. And lastly, before I turn it over to any questions, I do want to acknowledge Sitar and Simcyp. I've been fortunate to be part of the Simcyp, or the university academic licensing program now for about over 5 years. And I can't tell you how that's really dramatically impacted our graduate programs, our research, and really the graduates of our program now out working in industry at different companies, taking what they learned through this academic license program and applying it to drug development. I already mentioned Vamshi. He was really the architect of this study. And like I said, he's now employed at Gilead Sciences. Many of you may know Tycho Hanbach. He's been actively involved with our university, serving on thesis committees and visiting and interacting with our students. And he was instrumental in this particular project. And then Juliana is a colleague in our Division of Pharmacy Practice who was a co-author on the work. So with that, I'll turn it over, I guess, to you, Ali, and we'll take some questions.
Yeah, sure. So in terms of questions, there's the chat or the Q&A box. You can also raise your hand and we can unmute you if you've got any question that you're wanting to say.
I think Sibylla has her hand raised.
Oh, I haven't got down that far. Sibylla, can you speak?
Do we have to unmute her or?
Yeah,
oh no, I figured out I could unmute myself. Thank you very much. I really enjoyed that. And yes, and you uploaded actually the file, so that is also something I really appreciate. I think it is a great work that you actually found another CYP1A2 compound, because I see there really potential for building a matrix and getting therefore more confidence in actually the other comparts that we have in. You mentioned that you used for the rifampicin induction parameters, and you got that from literature value you mentioned. Can you say something to that? Because they are slightly different to that what we are using as induction inputs at the moment for rifampicin.
Oh, thank you, Sibylla. Yeah, I have the reference. It's in the publication that, this was, I think Vamshi built that model. That's why I don't know, maybe perhaps in the 2018 version that 1A2 parameters were not included.
Yeah.
I could send you the published reference that he relied on. There was a publication that on rifampicin 1A2, and that was what was used at the time to build those induction parameters.
Okay, thank you.
I'll send that to you. It's in the reference is in the publication. I noticed that as well when I was comparing the interaction parameters that we used with the current rifampicin substrate file that's in the Simcyp library. I noticed there's a difference there.
Yeah, I mean the difference is not that major, but there is a slight difference. So instead of your I think 3.8, we're using a 2.7, but the EC50 is about the same with a correction.
But I'll find out. I'll even check with Vamshi and get the information of maybe how he. I'll clarify that with him as well.
Yeah, thank you.
So there's another question from Jennifer from GSK. You should be able to unmute yourself now.
Hi, Ollie. Thank you. It's not so much of a question. I just wanted to shout out and say hello to Dr. Taft. Hi, Dr. Taft.
I didn't realize that's Jenny. Hi,
Yeah,
Jenny.
it's me. Hey. So sorry for people who are on the call. I was a student of Dr. Taft, graduated from LIU, and that's where I got my first hands-on with Simcyp when we had the first opportunity of the academic license. I'm now at GSK doing full-time PBPK modeling on Simcyp. So it was a long path, and I just couldn't stop myself from giving a shout out to you, Dr. Taft. So thank you for introducing it to me.
Well, thank you, Jenny. Yeah, Jenny's project was more. She did a lot of animal work for a company sponsor. So, but it's good to see that she's now, full on with her PBPK modeling. So thanks, Jenny.
Yes. Thank you.
I can't see any other questions right now, David, but obviously you provided your email address. So if there's any questions, people can contact you directly. Otherwise, can contact me or someone from Simcyp, and we'll forward on the questions to David. In terms of the next webinar in our series, we haven't announced this one yet, but please keep an eye out for that. The other thing just to highlight coming forward is there are a number of workshops that are going to be running online. So please, if you're interested in those, see them on the link there from the Learn Up website. I just want to thank David again today. It was a really great presentation. He explained it so clearly, and yeah, it's really good model, and thanks for your work in terms of putting this presentation together.
Well, Ali, thank you for the opportunity. I appreciate the opportunity as always, and I can't thank you and your team for all the support and interactions that we've had over the years, and I look forward to the continued partnership.
Yeah, great. Thanks again, David, and thank you everyone for joining us. Take care.
Thank you, everybody.
Bye-bye.